Welcome to the first part of Chapter 11. Chapter 11 covers cell communication. So this is the idea that especially in multicellular organisms, we have to coordinate what's going on. And we do that using cell signals. So these will be chemicals that disperse through our body to let us know what's going on. So for instance, if you have a water chugging contest, your body needs to realize that there's ultimately too much water. It needs to send a signal to your kidneys so they know that this idiot drank too much water. So we need to try to filter out some more water and dump it in the urine, so you have dilute urine to get rid of it. You could also have something where perhaps I decided to hit your fingers with a hammer, purely theoretical. But ultimately, if I did so, that signal of pain, of damage, has to be sent to your central nervous system to be processed, and then come back down so that your hand can respond by doing something like pulling away, or perhaps if you're a little bit more prone to violence, you could try to assault the person who's trying to hurt you. I wouldn't suggest that if it's me. But if that's just some of the examples with which that it's so important that our body is capable of communicating. Now, under that umbrella, we're going to discuss some of the generalities. And when we talk about cell communication, we'll very often talk about this signal transduction pathway. And this is just the general idea of what's going on. Now, the first part I've kind of added. We normally kind of cut this first part off of the signal. But something's going to happen. Some stimulus, something's going on that releases a chemical signal. This signal can come from the cell itself, or in some cases it could come from the other side of your body completely. So it doesn't really matter so much right now where it comes from, but we need this signal. And then where the magic starts to happen is if you have the receptor, and only if you have the receptor, you are able to bind that signal, oftentimes referred to as a ligand, you'll see. And so if that ligand binds to the receptor, it then transmits that signal. It causes a change inside the cell. And so this is what starts what we call transduction. And so this is where a series of changes happen inside of the cell prior to the final change, the final adjustment that leads to a response. And so once the transduction step reaches that final guy, which is oftentimes going to be like a transcription factor, which can cause a gene to be transcribed, which will cause it to produce the protein. So it's essentially turning a gene on. It could also block a gene to prevent transcription, which could turn a gene off. It could be an enzyme being activated or inactivated. But that'll be our response, is the actual effect that we get. But transduction is going to be this long, in some cases, convoluted process of step by step by step. It's like telephone, where somebody answers a door and gets the message. That's reception. But then you pass the message from one person to the next until it finally gets to the person who the message is intended for. And so that telephone part in the middle, that's going to be transduction. So those are the three general things: reception, transduction, and response. Now, when we talk about signaling, I kind of said before, ignore where the signal comes from, because it ultimately doesn't matter that much where the signal comes from. However, we have different names for kind of how long or how far away the signal's at. And so we will kind of discuss that a bit. But the general idea is a signal is a signal. And if it's received by a cell, the cell can do the response regardless of where it came from. But you will see that some cells will actually signal to themselves. That's called autocrine. So this is where the signal originates from a cell and affects that cell. So this one's kind of like you talking to yourself about what you should do. It can still make you do something. You can talk yourself into something. But ultimately, it's still coming from you and it's ending at you. You can also have paracrine signaling. This one's kind of like shouting fire in a crowded room. If I'm in another city, I could care less that you shouted fire because I can't hear it. I don't care. So things like growth factors, when your skin's damaged, can be released to cause that skin to start going through the process of cell division to patch up that area that was damaged. But it's only going to be released and it's going to stay in that area, so it only affects the cells that are nearby. So paracrine is going to be a relatively close distance thing. You'll also see sometimes that we'll talk about synaptic, which is where they're almost touching but not quite, so it's very specific from one person to the next. You can think of synaptic signaling as being a lot like when you've got a relay in track, where you're handing off the baton from one to the next. But imagine if for just like a split second at the end you kind of dropped it and then they grabbed it. So it wasn't a direct contact thing, because there is also a contact type of signaling, where like in our immune system it directly touches each other. So you've got autocrine from you to you. You've got contact where you actually touch. You've got paracrine, where you release it to everybody in the vicinity, but it does diffuse to them. And you've got synaptic, where you let it diffuse to somebody, but you're so close that it's very specific still. It's diffusing a tiny distance because you're almost touching, just not quite. This is like when you're a kid and you're like, I'm not touching you, I'm not touching you, and you're trying to get as close as possible, but not quite. And so synaptic will be like that, but it'll be very specific. So we've got autocrine, paracrine, synaptic. We can add contact here, or contact dependent it's sometimes called, where it's ultimately just going to be actually touching, and my handwriting is terrible. And then lastly, we've got endocrine. And endocrine is going to be the far distance. So that's the one that's kind of an oddball. You know, autocrine's kind of weird because you're signaling yourself. I mean, that's kind of sad. But you've got endocrine, which is going to be very long distances in many cases, where you're going to take a hormone that's produced in your brain, for instance, dump it in your bloodstream, and that hormone, like testosterone, can ultimately make changes start to happen far away in your body. You know, ultimately you can be talking about you've got chest hair now, or, you know, ultimately you're developing like the testes and things like that. So it's far away from the body. It's making changes happen. And so this one's typically going to go through the bloodstream. And so this will be a long distance signaling. So paracrine, even autocrine obviously is short distance because it's to yourself. Contact dependent, synaptic. A lot of these are going to be local, 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 really local. But it's going to be this idea of endocrine signaling, or hormones. That's going to be the one that's long distance. So this is you internet dating and whatnot, where you're doing this over long distances, where you can still get the signal there, but the signal's originating from oftentimes the brain, but far away in your body. It doesn't matter where it affects it, as long as the cell is in contact with that hormone that's in your blood, it's able to receive it, it's able to function. So this is how a lot of our hormones work, where they just dump it in the bloodstream, and a lot of cells will lack the receptor. So the cells that don't have the receptor just ignore it, but the cells that do have the receptor can use and express things based on that hormone regardless where they're at in your body. Now, our kind of example, our poster child, if you will, we've talked about a lot of these poster childs of signaling, is oftentimes going to be epinephrine. And so this one's going to involve epinephrine. And so when that signal is received, adrenaline's kind of the other name for it. When that signal is received, all right, what's going to happen here is it's going to activate what's called the G protein, all right? And so typically what happens there is this GDP gets replaced with a GTP. So we're essentially adding a phosphate, if you will, to this G protein is what we call it. So we've got this, you'll sometimes see it called the G protein linked receptor. That's just the receptor. And then this on the bottom here would be the actual G protein. And then once it gets activated, that G protein complex kind of breaks apart, and you can see this first subunit called the alpha subunit is going to just mosey on over to this other enzyme. And this other enzyme happens to be called adenylyl cyclase. Yes, there's two ills. I don't know if they just stuttered or whatnot, but ultimately it's what we do. And so once this adenylyl cyclase gets activated, its job is to take ATP and convert it into something called cyclic AMP. And the cyclic AMP is going to be used to activate other stuff further on. So it's ultimately going to allow some of these kinases, we'll have an aside here, a kinase is just going to be a protein that's commonly found in cell signaling that will, when a phosphate's added to it, typically allow it to add a phosphate to somebody else, which will activate them. So you'll see it's very common that we activate a kinase by essentially breaking an ATP and sticking a phosphate on it. And then it will oftentimes go ahead and go use an ATP to stick a phosphate on somebody else. And this process can continue in what's called a cascade, where oftentimes this happens with many, many different protein kinases, until eventually we activate the final guy, who in this case is going to be an enzyme that's going to convert glycogen to glucose. So we're going to depolymerize, is what this is commonly referred to as, glycogen. And that's what adrenaline does. Epinephrine's basically adrenaline. And the whole purpose here is if you're really freaked out, you want to make sure you're dumping simple sugars, things like glucose that we can break down for energy, into your bloodstream, because this is fight or flight. You know, we're assuming that this is all or nothing. And so your body is saying, screw the rest of digestion, here's some quick energy, don't die. And so this process of adrenaline in general is designed to make you as ready as possible. And that's why if you're driving somewhere and suddenly some random thing runs out in the road, I mean, it could be a raccoon, Girl Scout, whatever. And so if this thing runs out in the road, you might ultimately miss the person if you're not a very good driver. But the basic idea here is that regardless whether or not you hit it, you're going to be keyed up. You'll be strung out where every part of your body feels like it's vibrating because you've released this epinephrine. You've done these several different reactions. Your blood pressure is going up. You're having glucose be dumped there into your blood, so you're ready to go. You're basically becoming more or less, I guess we'll say, resistant to pain. So if you've ever been really scared and you're like running and stuff's hitting you and you just don't care, you can ignore it because your body is worried at that point about dying. It doesn't matter what it is, but for whatever reason, you've triggered this like severe, deep-seated reptilian brain kind of like survive process. And the end result is that you're ready to either fight for your life or run for your life, depending on your personality. And so this is one of the things that controls it. And you can see here how we've got all the characteristics where we've got the receptor, because it has to be received. If the cell doesn't have this receptor, because this would be in like liver cells, because that's where glycogen is. So if you don't have the appropriate receptor, then you would just ignore this. But if you have it, it's going to cause an internal change. In this case, it's that G protein that's connected to the G protein linked receptor. Not too difficult there. That's going to then be activated, and you'll see the activation is almost always by breaking an ATP, or in some cases, like here, a GTP. And by adding a phosphate, you typically activate it. Now later on, we can remove the phosphate, and there's a bunch of enzymes that are called phosphatases that will ultimately remove the phosphates and make these enzymes, these proteins, go back to their inactive state. And so that way we're not making these guys from scratch. We're just activating and deactivating them based upon our needs. Okay, protein phosphorylation, so kind of what we've been talking about. So the idea here is if you add a phosphate, you're going to activate the protein. And so that's what so many kinases will do, is they're going to add a phosphate to make the thing change its conformation and go into its active form, if you will. And so we'll call all the enzymes that do this, which they're proteins too, they're just called kinases or protein kinases. So these are the guys that usually themselves, once again, have to be activated. That's why it's oftentimes a cascade, where I get activated as a kinase, but then I also am able to catalyze the addition, the breaking of an ATP and sticking of a phosphate on the next kinase guy, which then activates him until eventually we get to the end. And we talked about these phosphatases reverse this, so it's not like once you get a chemical signal, you're stuck with it for life. We do have processes that get rid of this and make everything go back to the inactive state to make sure we don't keep acting like we're fighting or flighting for our life, whatever, forever. We want to make sure that this happens, but then after a bit we can cool down. So you will be twitchy for a while, but give it several minutes, I mean at most probably closer to like half an hour or something, and you should calm back down once the stimulus is gone. You can relax. And that's because we have these homeostatic processes to bring things back to the norm. We only want to kind of bump out of this temporarily. So if we have something that occurs, some kind of signal in this case is what we're using. We'll ignore some of the actual receptors and such, but that would exist. We're going to see this idea that you're going to activate this first protein by adding a phosphate, and then that's going to help catalyze the adding of the next phosphate to the second protein, so protein 2, and then protein 2 will catalyze the next guy having a phosphate added, which will activate him until eventually, in this case, we've got TP53. This is going to be a protein structure that will help determine whether or not a gene is used or not. And so in this case, if we add the phosphates to it, and you'll see there's several, one per subunit. Several of our proteins have multiple subunits that will allow this to become active and go bind to the DNA at a specific region, whereas otherwise it has these blockers, if you will, that prevent it from doing so. And so it's inactive. It's kind of just sitting around inactive unless this phosphorylation cascade kicks in, which ultimately activates it. And then this would be what causes our actual response, is this last guy who's not a kinase, who's able to go and attach and affect gene expression, or if it's an enzyme that actually does something. I mean, it could be an enzyme that's metabolic that will, once it's activated, start breaking down something, start producing something, and so this lets us control our body. You can also go through and amplify these steps because you could have one kinase activate several guys below him. And so the signal can snowball where one protein 1 might have been activated, but then that guy activates 3 protein 2s who can activate 10 protein 3s, etc., until eventually by the time we reach the end part, we've gotten this like snowballed cascade that now has lots and lots of protein kinases that it's dealing with. And so when it activates these end guys, it can activate many of them instead of just one. So we can get a bigger effect. And so we have this ability as the signal goes through transduction. This is one of the benefits of signal transduction, is that we can amplify or adjust the signal as it goes through this process, rather than just having it be 1 to 1 to 1 to 1, where the signal is fairly minor. We can have this be 1 to 100, so by the time we get to this last step, we have a much, much bigger signal that's present because we've amplified it. So there is a benefit, if you will, of the transduction step, is it does give you some more flexibility to control and manipulate that signal prior to the response to make sure we get the size response that we want. And then this idea is secondary messengers. So when we're talking about transduction, a common part of transduction isn't just kinases. We oftentimes too will have these molecules that are not proteins. So these are non-protein molecules that will act as a signal, if you will, themselves. And so we talked about, for instance, cyclic AMP. So if we convert ATP to cyclic AMP, the concentration of cyclic AMP can control stuff like these protein kinases later on in the process, if you will. So there's other guys too. There's IP3. There's a guy called DAG. You can have calcium. Whoa, that's supposed to be a G and that's supposed to be a C. So it's not like it's just cyclic AMP, but cyclic AMP will be one of our poster children as far as secondary messengers. And so it's kind of like you can just sit there as a cell, and if the concentration of calcium gets too high, it can trigger that phosphorylation cascade, or it can trigger an enzyme being altered in its conformation to be activated, so it can cause changes based upon the concentration of these secondary messengers. So this is another thing that we can use that's either in addition to or kind of in replacement of protein kinases to allow us to activate stuff. But it's just going to revolve around the concentration of these secondary messengers. So if you remember chemistry, we can do like the little concentration bars where that's the critical thing. You'll see this like with cell division, where if the concentration of cyclin gets high enough, it binds to another guy called CDK, which is a kinase, which then can trigger this, well, ultimately triggers MPF, but whatever, triggers ultimately your cell dividing. And so there's lots of these situations where it's not just like a pure on-off switch. It's more like that dimmer switch where at some point it's off, 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 but then it starts to kick on. And the more you typically turn this up, the bigger it'd be amplified, the more of a response that you would get. So it's a little bit more interesting where it gives you some more fine-tooth control. Just like we've talked about with the kinases, we have this kind of ability as well to try and, depending on what the kinase activity is, to try and amplify things a bit, scale it back. It's similar in that idea, except perhaps even a little bit more controllable, a little bit more diverse as far as how we use it and interact with it. So we've talked about epinephrine, and so just remember we talked about that enzyme that produces cyclic AMP, and so that's ultimately what's being activated first. And then it's that cyclic AMP that's produced that ultimately activates the rest of the process. So once we go through and we use GTP to activate this subunit, notice how this GDP switches to GTP, and it does that because we used a phosphate group from GTP. So one GTP donates a phosphate to a GDP, making it GTP. I know it's kind of weird, but you're actually involving two separate guys here. So GTP becomes GDP, and GDP becomes GTP. I didn't make this up, it's just what it is. And so once this process occurs, it activates the enzyme adenylyl cyclase, which then allows us to get cyclic AMP, and then this will trigger the later protein kinases that we talked about, if I could smell protein, which will then finish the process of making glycogen dump glucose into your blood. And then the cellular responses to signals, there's going to be several of these, but the basic idea is cytoplasmic activity. This is more or less turning enzymes on and off. So this will be control of enzymes. We have metabolism, which also is going to be, once again, adjusting enzymes on and off. It's just these enzymes are typically building or destroying stuff. Cytoplasmic activity could be moving stuff around. It doesn't have to be specifically destroying stuff or building it. transcription, which will be turning a gene on or off. So that one's not crazy. And then we're going to pick up with some of these guys with the next one, but it can also cause some additional changes as far as our immune system trying to make sure what cells are going to do. And it can be used in some cases for sex, because a lot of organisms you'll see don't have obvious things like eyes where they can look and say, hey lady. In many cases, like if you're a yeast, you're unicellular. You have no junk, if you will. And so they have to have some way of chemically saying, I am this gender. Genders really aren't male and female at that point, but they'll say kind of which gender I am, for instance, A or alpha, some examples, so that they can figure out if they should go through this mating process. So we'll pick up with this with the next podcast. I hope you guys enjoyed this. Have a good night.